Hi, welcome to the 3-minute lesson on heme synthesis. At the end of the session, you should be able to list the precursor molecules and tissues involved in heme synthesis, name the rate-limiting enzyme, and finally outline the synthesis of heme. Bone marrow accounts for nearly 85% of heme synthesis and liver for about 15%. Other tissues are also capable of synthesizing heme, but their contributions are smaller. There are totally 8 enzymes involved in the synthesis pathway, out of which 4 are present in the mitochondria and 4 in the cytosol. The starting materials for heme synthesis are succinyl-CoA and glycine. Succinyl-CoA is an intermediate in the TCA cycle and glycine is an amino acid. Heme synthesis also requires additional elements such as iron, zinc and vitamin B6. Let's look at an outline of heme synthesis before we begin. So we have succinyl-CoA and glycine to start with, which should be converted to a pyrrole. This pyrrole should then be converted into a porphyrin and heme. To be more specific, the joining of precursor molecules glycine and succinyl-CoA produces aminolevulinic acid or ALA. Two molecules of ALA then join to produce a pyrrole compound called porphobilinogen or PBG. 4 PBG molecules are then joined to form a porphyrin ring. This porphyrin ring is then modified and an ion is incorporated in its nucleus to complete a heme. The formation of ALA happens in the mitochondria and the reaction is catalyzed by an enzyme called ALA synthase. This reaction requires vitamin B6 as a cofactor. The first pyrrole compound PBG is formed from 2 molecules of ALA. This reaction is catalyzed by ALA dehydratase and zinc is a cofactor for this enzyme. 4 molecules of PBG then condense to give hydroxymethylbilane, which is a linear tetrapyrrole. This reaction is catalyzed by PBG deaminase or HMB synthase. This linear molecule is then converted into a cyclical ring called uroporphyrinogen or UPG by UPG synthase. UPG synthase will then undergo a 3-step modification on the ring to form protoporphyrin. The last step of heme synthesis involves incorporation of iron into the porphyrin nucleus. This reaction is catalyzed by ferrochelatase. The rate-limiting enzyme for heme synthesis is ALA synthase. In liver, major use of heme produced here is to be incorporated into cytochrome P450s, which are enzymes involved in metabolism of drugs. Here, heme acts as a repressor, that is, it decreases the synthesis of new enzymes. On the other hand, heme synthesis can increase with intake of certain drugs with use of the cytochrome P450s. That brings us to the end. Thanks for watching.